is also redox. So, most of the chapter I don't have you learn. I want you to learn the most important things about this. Almost all of you are going into health sciences, so we don't spend as much time on this. But we still give you enough so that you have a basic understanding of photosynthesis. And I feel like a traitor because I have worked with plants in research, and they're saying, chew on us? And I even talk about this anyway. So, photosynthesis. If it wasn't for photosynthesis, we would not be here. This is going to convert light energy into the chemical energy of bonds. So which law of thermodynamics is that? The first, that energy cannot be created nor can it be destroyed, but can be changed from one form to another. And that's what photosynthesis does. Now, plants are photoautotrophs, meaning they use light to produce their own food. We are heterotrophs, meaning we can't produce our own food. We have to ingest it from other organisms. Now, when the Earth was very young, the atmosphere was a reduced atmosphere, meaning it had a lot of methane. You know, more hydrogens on molecules, and methane is a carbon with 4 hydrogens. There wasn't carbon dioxide, and there wasn't oxygen as much. There was no oxygen, they think. They think it was mostly methane and your amino groups, ammonia. So very reduced atmosphere. And then you had cyanobacteria. Those are prokaryotes, but they're green. They're photosynthetic. And the thought is that the cyanobacteria produce the oxygen so that then other organisms could evolve that use oxygen. I'll play this at the end, the photosynthesis, this video. So photosynthesis occurs in plants and algae, protists. So when you were looking at pond water, you saw some of those little green things scooting around. They were eukaryotic organisms, single cell, with chlorophyll, with chloroplasts, and they were photosynthetic. So some protists are, and then again some prokaryotes. The cyanobacteria are prokaryotic cells, and they are green. Although they don't have chloroplasts. Their chlorophyll is embedded in the plasma membrane. Well, in membranes that go through the cell. It's invaginations of the plasma membrane. Now, again, we are heterotrophs. We depend on photoautotrophs for food and oxygen both. So, converting light energy to chemical energy, and then what do the mitochondria do? They convert the chemical energy to energy to drive ATP synthesis. Now, I didn't put a star on this because I won't ask it, but it's kind of nice to know at least about what leaves look like. But you do need to know that in the, you do have stomata, and that's where those gases, the oxygen is going to come out, and the CO2 is going to go in, into the leaf. And thylakoids are inside the chloroplast. Stroma is the dense liquid, so I do want you to know those 3 terms. So here if you have a plant, don't worry about the mesophyll or the mesophyll cells, but I do want you to know stomata. These are in the outside layer of the leaf, and they're just openings. These guard cells, they're 2 guard cells for each stomata, and they will plump up with water to close them, or pump water out of the central vacuole so that they shrink to open the stomata. And when the stomata is open, CO2 can go in and oxygen can come out. But also when it's open, water can go in and come out. And so leaves in tropical regions have to devise other methods to do a little different photosynthesis. Now inside the chloroplast, you have your thylakoids, which is a stack, and that's called a granum. And the thylakoid has a space in here. So you have the thylakoid membranes and the space in between. The chlorophyll is embedded in that thylakoid membrane, along with an electron transport chain and ATP synthase. Now, what is not pictured here, you have the inner and the outer membrane, you have your thylakoids, you have the stroma. What do we not see here that is here? DNA and, no, ribosomes. Yeah, mitochondria are separate. They're about this big, so they're not inside of them. But they do have ribosomes. This is an electron micrograph, and so you can see the stack of grain right there. Okay, so this is the one way to write photosynthesis, but usually I suggest that you do it the simpler way, and basically this is what most plant scientists say. 6 carbon dioxides plus 6 waters plus energy, which is light, yields C6H12O6 plus 6 oxygens. Now the difference here is that they've got water on both sides. So don't mix it up. Don't put 12 and then not put anything over here. If you're going to use this 12, make sure you have 6 waters over here. But this is, again, a simpler version of it. Because actually those 6 waters are kind of used in photosynthesis. And so a lot of times, most biologists, especially plant biologists, will just use this simplified formula of it. Now this looks like just the opposite of cellular respiration, doesn't it? But it isn't the reverse of it because it uses different chemical reactions and different enzymes. There are different processes. Although looking at just the formula, it looks like it is just the reverse of it. Now, 2 stars on this. Chloroplasts split water into hydrogen and oxygen and take the electrons away from it. So in cellular respiration, what was oxidized and what was reduced? Ooh, I should have asked that question.
Carbon is...
I'll ask it on the midterm.
Carbon is reduced, oxygen... No, oxygen is reduced, carbon oxidized. No.
Glucose.
Glucose is oxidized and O2 is reduced.
Yes, glucose is oxidized, oxygen is reduced. Now, in photosynthesis, it's the opposite. Water is going to be oxidized and CO2 is going to be reduced. Okay, so it reverses the direction of the electron flow when you compare it to respiration. Because in respiration, the electrons go from glucose to oxygen. In photosynthesis, it goes from basically oxygen to glucose. So it's the other way around. Now, photosynthesis is endergonic, cellular respiration is what? Exergonic. Okay, so the CO2 becomes reduced. Remember I told you that you usually look which gets the hydrogens? This is getting a bunch of hydrogens, so it is becoming reduced. NADH gets a couple of hydrogens, it's being reduced. Although you're adding the electrons, but if you want to just look at it easily and tell whether it's going to be reduced or oxidized, look at whether the hydrogens are added, because they have the electrons. And water becomes oxidized. Now remember, reduction is the gain of electrons, oxidation is the loss. So we're going to take the electrons from here away from there. Where in cellular respiration, this got the electrons. Now we're taking them away. Now there are 2 major portions, the light reactions and the Calvin cycle. Now sometimes this is called the dark reactions or the light independent reactions. These are light dependent, this could care less. Okay? So the light reactions split water, release oxygen, and they reduce NADP plus to NADPH. This is just like NAD plus, except it has a phosphate, another phosphate on it. NAD plus also has phosphate, but this has another one. So they're identical molecules except for that. And the ATP is going to be generated by photophosphorylation. So you've got substrate level, in glycolysis and Krebs cycle or citric acid cycle, you've got oxidative phosphorylation in the electron transport chain, chemiosmosis of cellular respiration. Now we're going to use photophosphorylation because that's where the energy is in the photons. Okay, so the thylakoids are going to transform light energy into the chemical energy of ATP plus putting electrons in it, NADP plus, and converting it to that. So which has more energy, NADP plus or NADH? NADH. It's a bigger molecule, more bonds, so it has more energy. So anytime you have more bonds, you're going to have more energy. So
that was NADH positive or
NADP,
Oh,
NADP plus has less energy than NADPH. So a reduced molecule has more energy in those bonds. Okay, so this, you just need to know this really well, okay? So you have water coming in. This is a light reactions. They take place in the thylakoids. Light comes in, NADP plus comes in, ADP and inorganic phosphate. You have ATP coming out, NADPH coming out, oxygen given off. The light reaction, or the Calvin cycle, takes place in the stroma. CO2 comes in, sugar comes out. And I put 3 stars on there. So you think maybe you should spend some time to memorize this? Yeah. Now I may ask you to draw it. I may ask you to fill in the blanks. I may ask questions about it. So any of the above. Yeah? Okay, so when a plant makes the sugar, it puts together 6 of those things. You know, so it makes G3, it makes glyceraldehyde 3-phosphate. And then it usually makes glucose out of that. And then the glucose, it will either store it as starch or put it into cellulose.
Also building.
Pardon?
Like growing.
Yeah. And also it will use some of the glucose in cellular respiration. Because plants have mitochondria because they have to produce a lot of ATP. The ATP that's produced here is used in the Calvin cycle, so it cannot be used in other cellular processes. All right, let's talk a little bit about light. So wavelength is the distance between 2 peaks or 2 troughs. Now, if you talk about the electromagnetic spectrum, you have everything from gamma rays clear to radio waves. The wavelength of gamma rays is 10 to the minus 5th nanometers. I mean, that's really very short. Radio waves are almost a mile long, so really long. Now, visible light is this little tiny portion right there. You have ultraviolet here, infrared, which we cannot see. However, insects, like bees, can see UV light. And if you put a UV light onto a flower, a lot of times you will see these little landing strip things that the bees can see, saying, oh, we're here, we'll go land there. And so they can see those. And they're patterns, so we don't know we're there unless you put UV light on it.
Is that why they go into those zapper lights?
Possibly. Of course, those are mosquitoes, they don't have a brain. Well, it's minuscule. I don't know, Florida's supposed to be getting these mosquitoes that are the size of a quarter. 4 of them can carry off a baby. I don't know. I'm going to Florida Friday. I'll let you know if I run into them.
Don't bring any home.
So my daughter, I said, okay, she wants me to help her plant flowers. I said, there are these things called snakes in your yard. She said, we don't have any snakes anymore. Florida got rid of all the snakes. Oh.
They have worms. They're just big.
Yeah. Okay, you have visible light, you have violet, and down here you have, you know, your red, everything in between. So which has more energy, violet or red?
No.
Purple. Violet, because it's a shorter wavelength. The shorter the wavelength, the more energy. Gamma rays kill you. They ionize your whole body. But radio waves, we have radio waves going through us right here. Doesn't hurt us at all because it's very low energy. So the higher the wavelength, or the shorter the wavelength rather, the higher the energy. Now, pigments absorb light. So if you have a purple shirt on, what is it absorbing? Everything but purple. Right.
Chloroplasts contains light-absorbing molecules called pigments. Different pigments absorb light at different wavelengths. One pigment, chlorophyll A, absorbs mainly blue-violet and red light. This molecule participates directly in the light reactions. Another molecule, chlorophyll B, absorbs mainly blue and orange light. We do not see these absorbed colors when we look at a leaf. Instead, we see the green wavelengths that are reflected back from the leaf or transmitted through it.
All right, so light comes through. So if we have green chlorophyll, it's absorbing what wavelengths of light? Everything but green. So the stuff on this side is green right in the middle. The stuff on this side and stuff on this side, but not right in the middle. Now, the absorption spectrum of chlorophyll A and the action spectrum. It's a little different, but they kind of spend some time to understand the difference there. This tells you which is going to be absorbed and therefore what's going to drive photosynthesis. And the action spectrum is the effectiveness of it. So if you look at this, these are photosynthetic pigments. Your chlorophyll A, which is bluish green, chlorophyll B is yellowish green, carotenoids are yellow and orange and kind of reddish. And so this is the absorption spectrum. Now, if you add these all together, you get the action spectrum. And so if you kind of draw a little line over all of this, doesn't that look like this? So all of these are contributing. And so one of the purposes of those accessory pigments are to broaden the spectrum of light that is effective in driving photosynthesis. It makes it more effective. Because if it was just chlorophyll A, which is your main pigment, that's not very much. But by adding these others, then you get more available work out of it. Now, why do you think that the action spectrum is higher here than here? More energy, exactly. Because this is the higher energy in, this is lower energy. So this is more, you get more work out of it on this end. So yeah, the peak is higher here than it is on the other one. Now, chlorophyll A, the main one, chlorophyll B is accessory. Now, the carotenoids also broaden the spectrum, but they also protect the leaf from too much light. So you've all seen these trees that have burgundy-colored leaves. If you grow those in shade, they are ugly. They're not burgundy because it doesn't need to produce those accessory pigments to block the sun. And I tried, I put a bush, one of these prune things, and it was in the shade, and I thought, oh, it'll work. It is ugly. I had to take it out because it wasn't burgundy, it was yellowish, ugly, yucky. So, anyway, don't try it, it doesn't work. I should have listened. Okay, this is what a molecule of chlorophyll looks like. Chlorophyll A and chlorophyll B are identical except for one oxygen difference. Now, in the lab, you did paper chromatography, right? Did it work?
Yeah.
It took a long time. It shouldn't have taken, usually it only takes about 3 or 4 minutes. Okay, so we're going to have a test tube here. And you're going to put a line of smushed up spinach right there. You had to be careful and get it really thick and not get it on the rest of the paper. And then you put a nonpolar solvent here. The nonpolar solvent went up through the paper. Now, what does a nonpolar solvent dissolve best? Nonpolar stuff. Now, the 4 pigments, photosynthetic pigments that are in spinach, you have chlorophyll A, chlorophyll B, you have xanthophyll, and you have beta-carotene. Now, what I always do when I teach labs is I hand out a paper that has the molecules of all of them, and I have everybody figure out what's the most polar and what's the least polar. Did your lab teachers do that?
No.
No, but anyways. You probably have lost those, because I had some that I gave, but that was about 5 years ago that I gave all the lab teachers those. You count the oxygens to see the polarity, because the more oxygens, the more polar. So how many oxygens are there in chlorophyll A? 5? How about chlorophyll B? 6. And I can tell you this much, this has 1 and this has 0. Okay. Actually, it's the other way around. Xanthophyll is 1 big here. All right, so this one is bluish green, right? This is yellowish green. Xanthophyll is lemon yellow. Beta carotene is kind of golden. Yeah, that's right. All right, so you should have had a thing up here that was shaped like that, because as it goes through, it's going to dissolve the pigments in here. Now remember, this is non-polar solvent. Then you have another band about there, and then right down here you have 2 bands that hardly separate at all. So what color did you get on top? Do you remember?
The golden.
Yeah, that's the most non-polar. So what should have been on the very bottom? The yellow. Yeah, the yellow-green. So if you remember right, did you get it to look like that?
Close.
You had to, in order to make it work, you had, first of all, you had to have really good solvent. And if the solvent has been open too long, it doesn't work anymore. And so you had to have really fresh solvent. And also you had to have a very thick layer of stuff, and you had to not get any speckles all the way through. And then it usually works out well. It only takes about 5 minutes at the most for you to really get that separation, that good separation. It's paper chromatography. Okay, so what we're going to do is we're going to make chlorophyll kind of get excited. So if you have energy coming in, you're going to excite those electrons, and then what happens to them?
They give off a red hue.
Pardon?
They give off a red hue.
Well, they go further away from the nucleus. They go to one of the outer energy shells that's empty. They don't like it out there. They're all alone. And so they want to come back down to where they belong. Now, there are 2 things when electrons come back down. They can either give off heat or they can give off light. In this case, they're giving off what? Light. And some heat too, but mainly light. Okay, why is it red? No, it's red. Less energy. If you have something that is green and it fluoresces, it will fluoresce at a lower energy level. So if you have something that's blue, what color is it going to fluoresce? Green. If you have something that's violet, what's it going to fluoresce? Blue. So it will fluoresce at a lower. That's just a bit of trivia, I won't ask it. Now you know why it fluoresces. It gets into light. So what I do when, in my research, when I would use green fluorescent protein, the light that I would use to excite it, the laser, I would use a blue laser, because then it's going to fluoresce green. So it will fluoresce at a lower level than it does. Okay, now photosystems. Just to mess with your brain a little bit more, if the redox didn't mess with it enough, we have two photosystems, photosystem 1, photosystem 2, and photosystem 2 happens first. It's because they discovered photosystem 1 first, even though it happens second. And they had too much pride, they wouldn't change. All right, so this is a photosystem, basically. And a photon comes in here from the light and pumps energy into the electrons. Now, the electrons move away from the nucleus, but they don't leave. Instead, when they drop back down, they pass that energy on to the next one, and then this one, and it keeps on happening until finally it gets here, and there's so much energy. These are the two special chlorophylls. There are no more special than those, they just happen to be there. And so they're called the special pair. It's really scientific. And now the energy is so much that those electrons actually leave and go to the primary electronic spectrum. Okay, so this thing sucks the electrons right off from these. So these two need to have those electrons replaced. So where do those electrons come from to replace them? Water. Remember, we're going to split water. We are going to oxidize water. So we're going to take those electrons and replace the ones that this guy sucked off from here. So the energy, this is energy, that these little arrows, they're not electrons, they're energy. And so this net has so much energy that those electrons actually leave and go up here. So this is the primary electronic scepter. And then water, again, is split to replace those. This is the thylakoid membrane. So kind of remember where we are, thylakoid space, that's inside. And then this is the stroma out here. Now, again, this is redox. And so that primary electron acceptor, since it's accepting electrons, is reduced. Now, two types of photosystems. Photosystem 2 happens first. Now, I did leave this in, and so where's there more energy? The next one has 700, the special chlorophylls. 700, this one is 680, so which has more energy? 680. 680, yeah, good. Now, two kinds of electron flow. We have linear flow and we have cyclic. And I do want you to know those differences. Now, linear flow, okay, here's linear flow. So the electrons leave here, go up to here, go down this electron transport chain. ATP is produced. So ATP synthase is in there. Then the electrons go over to here. This is another primary acceptor, down another electron transport chain. Now NADPH is the product there. So what is the final electron acceptor in photosynthesis? NADPH. So that's linear. The electron keeps on going and ends up on NADPH. So you have ATP produced, NADPH produced. Now in cyclic, the electrons do not go to NADPH. They just keep cycling around here, and you have ATP produced, but no NADPH. So that's the difference between linear and cyclic. Linear, both ATP and NADPH. Cyclic, only ATP. So keep that in mind for a little while. Now, I put 2 stars on this because I want you to understand the comparison. So, mitochondria, chloroplasts, you have electron transport chains and ATP synthase in both of them. And so you have a proton gradient. Now the darker color, more hydrogen ions, so the pH is what?
Lower.
Lower. Thank you. And so in the mitochondria, the protons are pumped into the inner membrane space. In the chloroplast, they're pumped into the thylakoid space. They come down through ATP synthase into the matrix of the mitochondria or into the stroma of the chloroplast. And this is a good question that I will be very likely to ask someday. That's why I put 3 stars on it. So really spend some time on that. Okay. Now let's go, this just shows that, so the NADPH and the ATP are going to the Calvin cycle. So all of the ATP that's produced by photosynthesis is used in photosynthesis. So the mitochondria have to do all the other ATP production in plants. Now, don't worry about this part. This is part of it, part of the Calvin cycle, but I won't ask it, but this you do need to know. Carbon enters the cycle as CO2, and then you have G3P, glyceraldehyde 3-phosphate, which exits. Also, if you just put sugar, I'll accept it. Okay. Rubisco is the most common protein on the face of the earth. So CO2s come in here. Now, they come in one at a time, and so to produce one G3P, you have to go through this 3 times. But these numbers are for 3 cycles. So to produce half of a glucose, how many ATP does it take?
2, 3,
9?
9.
How many NADPH is used? 6. So for a glucose molecule, how many ATP?
30 to 32. Oh, makes.
18. Because you have 9 for a half of a glucose, so you've got 18 for a full
So
glucose.
G3P is a half of glucose.
Yes,
Okay.
it's a 3-carbon sugar. Glyceraldehyde 3-phosphate. That's a lot of work to produce a glucose molecule.
Yeah.
Now, can you see why we have cyclic electron flow?
Yeah.
Because you need 9 ATP, but you only need 6 NADPHs. And cyclic just gives you ATP, so it keeps up. Now, I won't ask this, but it's kind of fun to know how plants in the tropics and in the deserts deal with the fact that if their stomata is open, they lose too much water. And so cam plants do it after midnight. They do the light reactions during the day, and then they incorporate CO2 during the night, and they can open the stomata when they don't lose too much water. And here it's the same thing, except they're going to do it in a different cell. But they, again, only open their stomata when they need to. Okay, so just a review here. And I have some questions for you. Maybe I should play that.
Yeah, you were going to play a video.
You're still going to do that.
I'm just taking up some more minutes.
I'm really tempted to not do the video.
Then just quiz us.
We only have 10 minutes.
Let's do the questions. Questions are better.
So we don't have to wait next time?
You are going to have a quiz next time, but this will help. Science will keep away.
So the next chapter is 11 and 12, and this number 11.
The term?
Yeah.
It'll be at least through 12 and maybe 13. So it'll be 9, 10, 11, and 12 probably. Okay, without looking. What's this?
H2O.
H2O in this?
Light.
Here?
Light.
Light reaction. Light reaction. This? Oh, by the way, what's this?
O2.
O2. Yeah. NAD plus, ADP plus inorganic phosphate,
ATP. Okay.
oxygen. And up here, CO2, Calvin cycle, and sugar comes out. Half of sugar. This is a sixth of one, actually. So one cycle gives you this. So to get glucose, you've got to go through 6 cycles. Okay. I'm hoping we can get through all these questions, because the last question's really good. Well, that whole thing that with all the chlorophyll in it is the antenna. It's chlorophyll plus a lot of protein. And it's in the photosystem 1. Photosystem 2, rather. Actually, it's called Photosystems.
I'm like writing it down.
Hey, what do you think? Well, we know it's not A because oxygen doesn't go to the reaction. No. Electrons do. Synthesize ATP from no. Transfer electrons to paradoxin and then NADPH. No, that happens in the electron transport chain. Harvest photons and transfer light energy to the reaction center chlorophyll.
Yeah.
That sounds good. But thank you. Well, you've got ATP synthase in chloroplasts, you have ATP synthase in mitochondria, and it's proton gradient that powers both. Okay, splitting of carbon dioxide to form oxygen gas and carbon compounds occurs during. Be careful.
Oh, neither.
Yeah, water split, not carbon dioxide. Carbon dioxide is glued together to make glucose. Okay? This is a really good one because this says whether or not you understand both chapter 9 and 10. So you have a flask with green algae and a control flask that just has water with nothing. They're both placed in a bank of lights, which cycles between 12 hours of light and 12 hours of dark. The dissolved oxygen concentration in both flasks are monitored. So, predict what the relative dissolved oxygen concentrations will be in the flask with algae compared to the control flask. So is the control flask going to change? No. Okay, so just your test flask will. Let's vote. Alpha. Beta. Chi. Delta. Epsilon.
Fully agree.
Except I said something about chapter 9, didn't I? Remember, plants have mitochondria. They're going to be doing cellular respiration.
Was I right with A?
No, because does cellular respiration release oxygen or use it? It's released.
No, it's using it. It's using it. So the carbon dioxide.
Glucose plus 6O2 yields water plus CO2.
Okay.
So it's using it. So it's not C. So what do you think it is?
D, delta, delta.
Oh, the,
Yes. Because in the light, the plants are going to be producing oxygen a lot, more so than they're going to be using. But in the dark, they don't produce oxygen, they just use it. And so it's going to have more oxygen than the control flask in the light, less oxygen than the control flask in the dark. It is a good one. Actually, we'll have time to do the